Dear students, I welcome you all to the 18th session of Reproductive Biology and Developmental Biology paper prescribed by the Mangalore University for 6th semester BSc Zoology. So in this session, we are going to study about a detailed aspects of cleavage. The fertilization results into the formation of zygote. In the previous session, we have seen the different aspects related to the process of fertilization, how the gametes approach, how the sperm enters into the egg, what are all the chemical reactions involved, what are all the physical issues involved in that, and how the sperm enters into egg, and how pronuclei are formed, how they fuse, how they produce zygote. Everything we have studied in the previous session. So now, what after the formation of zygote? Once the zygote is formed, the development is initiated. How the development proceeds? What are all the factors that are involved in the process of development? And how the development actually transforms a single cell zygote into an entire animal is the focus of our study in the coming classes, including this session. So now here in this session, let us see how the single celled zygote becomes a multicellular structure and then becomes a blastula and gastrula and so on. The process of segmentation, that means cleavage, immediately follows the fertilization or any other process which activates the egg. Cleavage consists of several mitotic divisions of the zygote into a large number of cellular entities. The cells which are produced during the segmentation are called blastomeres. So this kind of mitotic division is quite different from, it's quite similar, not different, it's quite similar to the mitosis that takes place in other somatic cells as well. But instead of calling these daughter cells as daughter cells, we call these resultant products of the mitosis here as blastomeres. Mere referring to a piece. At first, the cells remain closely associated in the beginning, but later on, as the development proceeds, they form the lining of a hollow sphere called blastula. The blastula contains a cavity named as blastocele, and its outer covering is designated as blastodome. As you can see here in this photograph, the zygote proceeds through a series of mitotic divisions and forms a mass of cells having several cells in multiples of 2. And ultimately they form a sphere having a cavity inside, and this is called a blastula stage. The process of segmentation prepares the groundwork for the future design of the embryo by producing adequate number of cells. So in order to produce an entire embryo, the basement will be formed by the blastula. The cleavage also establishes the fundamental conditions for the initiation of next developmental stage, the gastrulation. Before going into the details of cleavage, Let us see some of the important laws that were framed long ago in 17th and 18th centuries, some in 19th century as well, which clearly describes the behavior of the cleavage. And people observe that the patterns of cleavage is almost uniform across the animal kingdom. So, as a part of it, we can generalize the patterns of cleavage as well as the way how cleavage converts a zygote into a blastula. Let us first look into Sachs' law, which he proposed in 1887. This law consists of two postulates. The first postulate being the cells tend to divide into equal daughter cells. Though this statement looks a bit awkward today because we know that the cleavage plane does not divide the cells into two equal daughter cells always. If you observe the third cleavage of amphibian egg, you can see here the third cleavage, which is latitudinal. The plane is just above the center of the egg. It divides the blastomeres. Blastomeres means the daughter cells into two categories. The smaller one are called micromeres and the larger one are called the macromeres. This is just based on the size. But this third cleavage may look an exception for this statement, but in 1887 the statement was produced. So by observing the first few cleavages, which you can see here, the meridional one, the one which cleaves the ovum or egg from the animal pole towards the vegetal pole, you can see that this plane is cutting the zygote into two daughter cells of equal size. The second statement being each new division plane tends to intersect the preceding plane at right angles, which acts to maintain the spherical shape of the blastomeres. So this is correct, and still today this law is this particular postulate of the law is accepted widely, because you can see here the first cleavage, which is meridional, the one which is cleaving the zygote from its animal pole towards the vegetal pole. If you look at the second one, it is cleaving the zygote from the animal pole and towards the vegetal pole. But the plane of cleavage, though it is meridional, it is exactly perpendicular to the previous plane of cleavage. And if you look at the third one here, though it is latitudinal, though it is producing the blastomeres of unequal size, this plane of cleavage is bisecting the previous planes of cleavage at exact right angles, right? So same way happens in almost all cleavages further. Let us look into the next law of cleavage, which was given by Hertwig in the year 1881. In 1881, Hertwig added few laws of cleavage which were earlier described by Sachs. This rule emphasizes the cell shape to be a default mechanism of spindle apparatus orientation. Hertwig's rule predicts cell division orientation that is important for the tissue architecture, cell fate, and morphogenesis. That means the division plane of the cell, how the cell divides, in which plane it divides and how it produces the blastomeres is the key factor that determines the architecture of the tissues. That means the future tissues, as well as cell fate, the fate of the cell, what the cell is going to become in the future, as well as the morphogenesis, the genesis of organs. In his experiments, Hertwig's, the orientation of divisions of the frog egg was studied. Frog egg has a round shape and the first division occur in random orientation. Now Hertwig compressed the egg between two parallel plates. The compression forced the eggs to change its shape from round to elongated. Now Hertwig noticed that the elongated egg divides, but not randomly, but orthogonal to its long axis. The new daughter cells were formed along the longest axis of the cell. That observation later got the name Hertwig's rule or long axis rule. By definition, this rule is the nucleus and the achromatic figure. Achromatic figure means the mitotic spindle. Occupy the center of the protoplasmic density of the egg or blastomeres in which it lies. Hence, in microlycisal and isolycisal eggs, the spindle is located centrally. In telolycisal ovum, it is nearer to the animal pole. See, if you look at these photographs, the spindle is dividing the cell exactly orthogonal to the longest axis. So this is the longest axis of this ovum now, and it is bifurcating these cells here. Same thing happens to the next cell. See, now the second cell, what happened? This became the longest axis, right? This, not this one. So now what happens? Now earlier, so when this was the single cell, I will show you. Now this is a single cell, which is the longest axis. Now this is the longest axis, right? So now what happens? The cleavage goes like this. Now second cleavage the same way, right? So it goes like this. This becomes the longest axis. Now achromatic figure, that means the nucleus will be exactly situated at the center here of the protoplasmic density. What is protoplasmic density now? Protoplasmic density is nothing but cytoplasm, not the yolk. Cytoplasm excluding the yolk. That's why what happens if you observe the ovum of the frog after fertilization, let us say zygote of the frog, the nucleus will be situated somewhere here, just above the center, not exactly at the center, but just above the center. But if you observe the ovum of the chicken or any reptiles, you can see that the nucleus and the achromatic figure situated in this position, exactly where there will be a blastode disc. This portion will be completely filled with yolk, right? So the nucleus will not be there here, and it will not bifurcate this yolk. It only cleaves this part, which we consider today as blastode disc. Let us look into next two laws of the cleavage. The third being the Balfour's law. Balfour's law was formulated by Balfour in the year 1888, and it states that the speed or rate of cleavage in any region of the egg is inversely proportional to the amount of yolk or deutoplasm it contains. That means in telolacetial egg, blastomeres in the animal pool divide faster than those nearer to the vegetal pole. This you can clearly observe in the eggs of chicken. See, this is the view from the upside of the cleaving chicken egg. Or let me give you one more example of the ovum of the frog, the zygote of the frog. See, what happens in zygote of frog is the deutoplasm. What is deutoplasm? Deutoplasm means the yolk, the one which is written in yellow color here is, okay, sorry, this portion will be yolk. I'm writing it in yellow color just to show you that yolk will be in yellow color. Okay. Now this part will be now full of yolk. Okay, and now this part is the animal pole. I will write it as AP. Okay, now this is the vegetal pole. Let me write it as VP. Now what is the core of Balfour's law A? Balfour's law states that the speed or rate of cleavage is inversely proportional. That means if the amount of yolk is high, the speed or rate of cleavage will be lower. If the amount of yolk is lower, then the speed of cleavage will be higher. So this you can clearly see after the third cleavage in frog eggs, which we are going to study in detail in coming sessions. Okay. Here what happens? The first cleavage will be fine. Okay, the meridional one bisects like this, second one goes like this, the third one starts from here now. So this third plane of cleavage will clearly differentiate the entire zygote into two parts. Okay, the upper part having smaller cells near animal pole, the lower part having larger cells near the vegetal pole. Now once this cleavage is over, the next cleavages will be faster in this region, just because they do not contain any kind of yolk, or the quantity of yolk is very less. And the rate of cleavage in this portion will be little slower than this. So what happens? The cells in this region after 32 cell stage or so, you may find larger cells in the blastula of frogs, which I will show you later on. Okay, like this. And in the animal pole, what happens? You can see only small cells like this, which are now micromeres. So now division of micromeres will be faster than the division of the macromeres. This is just because of the presence of the yolk. And the last law which we are going to study here is the Flugus law, which is defined as the mitotic spindle always elongates in the direction of least resistance. That means the mitotic spindle. Okay, what is mitotic spindle now? Mitotic spindle means the cell division mechanism. machinery. Fine. Now let me show you this. So imagine this is a zygote. Let us consider this is the nucleus. Now you will have to have centrioles, okay, two centrioles, which will produce numerous asteroids like this, right, which will bifurcate the cell. Now this machinery, that means these asters, will drag the cells, elongate only in the portion where there is least resistance. Least resistance means the cytoplasm which is not containing yolk. See, yolk is the major disturbance to the cleavage. Always the yolk tries to stop the cleavage because the cleavage plane cannot pass through the yolk. That's why what happens in amphibian yolk, the first cleavage will be like this. Okay, here there will be a lot of what is that, yolk. So what happens? These spindles elongate in this direction. That's why what happens when the cell division is there, the plane of cleavage you can observe like this. So now they bifurcate this, the cell will be bifurcated in this meridional plane. Okay, this is how you can explain the Fluger's law. I hope you have understood the laws clearly. Now I will move on to the next very important topic, the planes of cleavage. During early cleavage, a distinct geometrical relationship exists between the blastomeres. That means each plane of cell division bears a definite relationship with each other. Based on the way how the planes of cleavage bisect the zygote, we can categorize the planes of cleavage into 4 different categories, types. Now the first type being the meridional cleavage, meridional plane of cleavage. Now what is a meridional plane of cleavage? When a furrow bisects both the poles of the egg, passing through the median axis or center of egg, it is called meridional cleavage. The median axis runs between the center of animal and vegetal pole. Now imagine now this is the nucleus, and if the plane of this is animal pole, you know that, right? And this is vegetal pole. Anyhow, in this diagram you can clearly see that. Okay, this is vegetal pole, this animal pole. Now you focus on this plane only, don't worry about other planes. Now carefully observe this plane. This plane is bisecting the zygote or blastomeres into 2, and it is passing from the animal pole towards the vegetal pole. So this kind of cleavage plane is called meridional plane, but the important factor here is it has to pass through the center. It has to pass through the center here. Now the second plane being vertical. The vertical plane of cleavage is when a furrow passes in any direction, but it does not pass through the median axis from the animal pole towards the vegetal pole. It is called a vertical plane. You carefully observe here. This is the vertical plane. It is not passing through the center. If it passes through the center, it is called meridional plane. If it is not passing through the center, but anyway If it is passing from animal pole towards the vegetal pole, then it is called a vertical plane. The third being the equatorial plane. So this equatorial type of cleavage plane divides the egg halfway between the animal and vegetal poles, and the line of division runs at right angles to the median axis. So look at this plane now. Okay, exactly at the center. It is equatorial. That means it is passing halfway through the animal poles and vegetal pole. Exactly at the center, passing through the central axis, median axis. Median axis means the central axis. Okay. Now it is passing through that median axis or central axis. Fine. And then such a type of cleavage plane is called equatorial plane. So next plane is the latitudinal plane of cleavage. So this cleavage is almost similar to the equatorial plane of cleavage, but the furrow runs through the cytoplasm on either side of the equatorial plane. So if you can see here this plane, the transverse plane, which is also called the latitudinal plane, that bisects the zygote from right to left, anywhere above or below the equatorial plane. So please remember, if the plane passes through the central axis and if it is bifurcating the zygote from animal pole to vegetal pole, that is meridional plane. If the same plane is running right to left, passing through the central axis, it's equatorial plane. If the plane is vertical and if it's running from anywhere beyond the median plane, then that is vertical. Or the plane is latitudinal or transverse if it's moving from anywhere above or below the equatorial plane. Now let us see, based on the way how the cleavage cuts the zygote, there are two types of cleavages, the holoblastic cleavage and the meroblastic cleavage. Now, when you look at the types of cleavage, a considerable amount of reorganization occurs during the period of cleavage, and the types of cleavage depend largely upon the cytoplasmic contents as well. The amount of yolk, the lecithality, also determines the type of cleavage, which are as follows. The holoblastic cleavage. Holoblastic cleavage is a major pattern of embryonic cleavage or embryonic cell division, in which cytokinesis completely separates the cells during division. Generally, the eggs like alycetal eggs, homolycetal and mesolycetal eggs show rapid and complete divisions of zygote and are called total or holoblastic cleavages, resulting in 8 blastomeres after the third cleavage, may be equal or unequal to each other. So accordingly, there are two types. You carefully observe this now, this plane, okay, of cleavage here. It's running from this part, the animal pole, towards the vegetal pole, and it is completely bifurcating the zygotes, producing 2 daughter cells, right? So this kind of cleavage is called the holoblastic cleavage. Now, based on the types of blastomeres produced, there are again 2 types. Let us see which are they. The equal holoblastic and unequal holoblastic. If the formed blastomeres are approximately equal in their size, then such cleavages are called holoblastic equal cleavages. You can see in this photograph, the cleavage is producing two blastomeres which are approximately equal in their size, both the sides, right? Even in the 4-cell stage, the cells looks almost similar, right? So this type of cleavage is called equal holoblastic cleavage, which is observed in the zygotes of echinoderms, amphioxus, as well as in placental mammals. The unequal holoblastic cleavage. Now, if the formed blastomeres are unequal in size, then such cleavage is called unequal holoblastic cleavage. The resulting upper 4 blastomeres are generally smaller, and the lower yolk-laden 4 yolk-laden larger blastomeres. You can see here the third cleavage in amphibians is generally unequal holoblastic cleavage, where you can see this cleavage furrow, the third cleavage furrow, is bisecting the zygote just above the equatorial line. If you draw a hypothetical equatorial line, this will be somewhere here. Now just above this you can see a plane of cleavage which is bisecting. So it is producing two blastomeres of unequal size. So such a type of cleavage is called unequal holoblastic cleavage. If you move further, the next type of cleavage is called the mirroblastic cleavage. In large polylecithal eggs, such as the eggs of reptiles and birds, the cleavage furrow cannot cut through the enormous yolk present, so that the entire egg is not divided into cells. Thus, the cleavage is incomplete or partial and is termed as mirroblastic, and it's of the following two types. So if you see here, if you consider this as an ovum, and let us consider this as the egg of a bird. Okay, so you know that the egg of bird is a polylecithal one. It's having lot of yolk like this, right? And then fine, just above that you will find a germinal disc or blastodisc. So now the entire process of cell division must take place in this particular part. Okay, the cell division or the cleavage is confined to this part, right? So now the furrow cannot divide. Let us say, for an example, if the furrow is dividing in this way, you can see here a cleavage furrow in the bird's ovum. Now this will not bifurcate the entire ovum. Only the division takes place in this region. This furrow will not move through the yolk and divide the egg. So this is not possible in a polylecithal egg, because the yolk will not allow this furrow to pass through. Hence such a kind of cleavage is called mirroblastic cleavage. Now it's of two kinds. The first kind is the discoidal cleavage. That means the cleavage is restricted to only a small cytoplasmic cap at the animal pole, resulting in a rounded embryonic or germinal disc, and is termed as discoidal cleavage. This is what I am explaining you. Okay, this is the egg of a bird, or in reptiles the same way happens. Okay, the cleavage furrow will not bisect the entire egg, just this part will be, see, this is the active part, and only this part will be divided during cleavages. Okay, so hence since the cleavage is restricted to a disc shaped structure, cytoplasmic structure above the the yolk, this kind of cleavage is called discoidal cleavage. The next type is the superficial cleavage, where the cleavage is restricted to a superficial peripheral layer of cytoplasm around the yolk, and hence the term superficial. So if you can see here, the cleavage is restricted to the periphery. The entire egg will not divide here. What happens is this is the developing stages of a arthropod egg. Now you can see here a single nucleus is there in the egg, and when the fertilization happens, all the nucleus present in this divides into several nuclei, okay, many pieces, and each piece moves towards the periphery, and surrounding that particular piece of nucleus, the cytoplasm and the septa like structure, the plasma membrane appears, forming a structure which is called syncytium or superficial blastula. So this type of cleavage is considered as superficial cleavage. Superficial means the egg will not divide, only the nucleus will divide. Superficial ಅಂದ್ರೆ ಮೇಲ್ನೋಟಕ್ಕೆ ಅನ್ನೋ ಅರ್ಥ ಬರುತ್ತೆ. ಇಲ್ಲಿ ಸಂಪೂರ್ಣವಾಗಿ ಮೊಟ್ಟೆ ಏನಿದೆ, ಎಗ್ ಏನಿದೆ, ಡಿವೈಡ್ ಆಗೋದಿಲ್ಲ, ಬದಲಾಗಿ ಏನಾಗುತ್ತೆ, ನ್ಯೂಕ್ಲಿಯಸ್ ಡಿವೈಡ್ ಆಗುತ್ತೆ. ನ್ಯೂಕ್ಲಿಯಸ್ ಡಿವೈಡ್ ಆಗಿ ಆ ಎಗ್ ಏನಿದೆ ಅದರ ಸುತ್ತಲೂ ಹೋಗಿ ಸೇರಿಕೊಂಡು ಅದರ ಸುತ್ತ ಸಣ್ಣ ಸಣ್ಣ ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಡೆವಲಪ್ ಆಗಿ ಎಂಬ್ರಿಯೋ ಮುಂದೆ ಡೆವಲಪ್ ಆಗ್ತದೆ. ಬೇರೆ ಸಂದರ್ಭದಲ್ಲಿ ಆಂಫಿಬಿಯಾದಲ್ಲಿ ಏನಾಗ್ತದೆ, ಈ ಎಗ್ ಸಂಪೂರ್ಣವಾಗಿ ಡಿವೈಡ್ ಆಗ್ತದೆ. ಹಾಗೆ ಡಿವಿಷನ್ ಇಲ್ಲಿ ಇಲ್ಲ, ಎಗ್ ಡಿವಿಷನ್ ಇಲ್ಲ, ಓನ್ಲಿ ಇಟ್ಸ್ ನ್ಯೂಕ್ಲಿಯರ್ ಡಿವಿಷನ್. ಹೆನ್ಸ್ the name superficial cleavage. All the arthropods are the examples of the eggs having this kind of cleavages. Now there are other types of classification of the cleavage plane as well. The radial cleavage is an example, wherein the cleavage plane cut the zygote in such a manner that there appears a radial symmetry in the resulting blastomeres. So such a type of cleavage is called radial cleavage. Now I hope you remember what is radial cleavage, radial symmetry. Let us consider this as a coelenterate which exhibits radial symmetry. See, radial symmetry means if you cut the animal through any plane, you are supposed to get two equal halves, right? That is radial symmetry. Now exactly the same way, look at this structure now. This is the way how the blastomeres are arranged in the radial symmetry. You cut this through any plane, you are going to get two equal halves. So this is the result of the radial cleavage. Okay. For an example, the frog's zygote divides by a vertical furrow into two equal blastomeres. The second cleavage furrow is also vertical, but appears at right angles to the first. Thus, four blastomeres are produced. The four blastomeres remain sticking together, just like this. Okay. Now, a horizontal cleavage then appears above the equatorial region to cut the four blastomeres into 8 blastomeres, with 4 smaller upper blastomeres and the 4 bigger lower blastomeres, just like this. See, this is how it will be initially. 4 blastomeres are produced, and now one cleavage furrow cuts like this way, so the upper ones become micromeres and the lower ones become macromeres. But at this stage, each bigger blastomere has a smaller blastomere sitting on its head, and the blastomeres are arranged in 4 radial planes. Just like this, you can see that there will be a megamere or macromere below, above which you can find a micromere, okay, and all are arranged in a radial axis just like this. This is what the radial cleavage is. Just remember, radial cleavage is the one where you can observe the radial symmetry when the cleavage happens. Go to the second type, which is the spiral cleavage. In spiral cleavage, it is found that those forms in which there are rotational movement of cell parts around the north and south pole axis of the egg, leading to a displacement or inclination of the mitotic spindles with respect to the symmetrically disposed radii. Here the cleavage planes are neither vertical nor horizontal but are slanting in relation to this axis. Moreover, each blastomere divides to form one bigger cell and a smaller cell. In such a cleavage, the blastomeres of upper tier, the micromeres, sits over the junction between each two of the vegetal blastomeres, which are macromeres. This is due to oblique position of the mitotic spindles. Therefore, this is also called as oblique cleavage. In successive cleavages, the mitotic spindles are arranged in a sort of a spiral. The turn of the spiral may lie in a clockwise direction, which is dextral cleavage, the right-hand one, or anti-clockwise direction, the left-handed, which is also called a sinistral cleavage. These types of cleavage could be observed in turbellarians, nematodes, rotifers, annelids, and all mollusks except cephalopods. Now carefully observe here, the spiral cleavage is nothing but when the cleavage furrow bisects the zygotes into different, many daughter cells, blastomeres. In spiral cleavage, what happens? The micromeres are produced exactly the same way how they are produced in the previous radial cleavage. But here what happens? In radial cleavage, the micromeres were exactly sitting just above these macromeres, right? Immediately above that. But here what happens? Here the micromeres sit like this. So they are situated in between the two macromeres. Now what happens? See, if the nucleus of this macromere is here, this nucleus is here, this nucleus here. Now what happened? Oblique line can be drawn here, right? So the daughter cells are produced in this fashion, right? So what happens? There will be a central axis around which you can find the cells just moving like this. They look like a spiral. Hence they are called spiral cleavages. To make it very clear, I will also write how the radial cleavage will be. See, in radial cleavage, this is how it will be. Fine. Two blastomeres produced. Okay. And exactly above which you can see their micromeres. Like this. But, so there will be a straight line between these two. But what happens here? You can see a oblique line like this in this spiral cleavage. So this is the difference between spiral cleavage and the radial cleavage as well. For any further information about the cleavages and types of cleavages, you can refer these textbooks. Thank you.